you. Hi guys, my name is Tania. I'm a member of the iGEM Measurement Committee. I have also been a judge at iGEM since 2018. And today I will talk to you about some of the basic techniques in molecular biology. The first technique is the polymerase chain reaction, which is commonly referred to as PCR. PCR is the fundamental technique used to make copies of gene sequences. These PCR products are commonly referred to as amplicons. Moreover, metabolic engineering requires DNA manipulation using PCR. Understanding how PCR works is essential for developing new DNA-based technologies. Because of this, we encourage you to watch the link video that will explain how PCR works. The second technique is restriction digestion, where digestion of DNA happens using restriction enzymes. Briefly, polymers of double-stranded DNA are cleaved using restriction endonucleases that recognize specific nucleotide sequences. This technique is a standard procedure for many molecular biology workflows, such as gene cloning, where circular plasmids are cleaved by restriction enzymes in order to insert PCR products such as protein coding sequences into the plasmid. This video explains the mechanism in more detail. In contrast to restriction digestion, there are also molecular techniques that join separate ends of DNA together. A common way to do this is using the DNA ligases, where amplicons from PCR are inserted into open vectors to form recombinant plasmids using DNA ligase. To do that, DNA ligase forms phosphodiester linkages between the ends of the amplicons and the open ends of the plasmid. As you will hear in the video, describing how this process works. A common DNA ligase used for gene cloning is T4DNA ligase, an enzyme that was found in a bacteriophage. The final basic technique we want to bring up today is bacterial transformation. This is when bacterial cells are transformed by taking recombinant plasmids into the cell. Bacterial transformation occurs via chemical transformation or electroporation, two methods that are described in the video. The transformed bacterial cells are cultured on selective medium corresponding to selection marker gene expression, which is generally encoded on the recombinant plasmid. Thanks for listening. I would like to, once again, I highly encourage you all to watch the videos of these basic techniques for gaining more depth understanding. And also, I would like to remind you that we have more upcoming webinars on exciting topics such as modeling circuits and DNA assembly techniques. I hope you all have a wonderful rest of the week, and thank you for your attention.